Thank you all for being here. This is, whenever I drive up here, I'm always amused that this is officially the U.S. Postal Service training facility. So we do aim to do assays come rain, sleet, snow, or dark of night, and perhaps that's why Nathan decided to have the session here. What I want to do just briefly is put in context what you're going to do for the next day, and that is I want to start off with what Nathan talked about, which is what NCATS mission is and how this manual fits into it. So you're all familiar with the fact that fundamental science has really exploded in the last 30 years. When I look back to when I finished training, now over 30 years ago, compared to then to now, it really is a different landscape. However, if you're a patient waiting for treatments, potential treatments from that explosion, for the most part, you're still waiting. I'm a neurologist by training, and my world in clinical neurology is not, for the most part, appreciably different than it was 30 years ago, even though the genetics of neurological disease, for instance, is quite different. And so this problem of going from this explosion in fundamental discovery to treatments that are shown to affect human health, this is the process that we call translation, of course. And NCATS was formed about 5 years ago to work on this problem as a scientific problem. Those of you who are practitioners of this will know that for the most part, translation has been an empirical trial and error process, the rules of which, the fundamental principles of which have never been studied for the most part. It's never been studied as a scientific problem. And as a result, whenever one has a problem like this that has an increase in number of degrees of freedom over many years or decades that this process takes, and one is taking what is for the most part a relentlessly empirical approach, the likelihood of your getting successfully to the beginning to the end with that trial and error approach is zero, and in fact that's exactly what we see in this process. And so our feeling is that the only way to make this process more efficient and effective is to understand what the general principles are, just like any other area of science or engineering. And so when NCATS was formed 5 years ago, almost 6 years ago now, its purpose was to start that area of science in a robust way. And you'll notice that we use words like catalyze, innovative methods and technologies, new ways of doing things that will get treatments to patients better, faster, cheaper. And so this is our definition of translational science. It's the field of investigation that seeks to understand what the general principles are by which this process happens. And it's important that even more than other areas of science, this is both a scientific problem and an organizational problem. I mean, if you look at the faculty of today, they come from an extremely broad array of entities within the biomedical research ecosystem, and that's absolutely typical. But what it means is that, as we like to say at NCATS, that translation is a team sport, and so what that means is that it needs to be run in a quite different way than a typical single investigator, single postdoc in the lab fundamental science. And that, again, that model works great for fundamental science, but doesn't work at all for translation. And so it means things like credit for team science or collaborative structures can be limiting in this process, whereas it really isn't in other areas of science. So what are some of the problems that we work on? Well, this is those of you who work in this space will will be familiar with this rogues gallery of problems that bedevil every translational project, no matter whether one's working on Alzheimer's disease or eczema. These are all the same problems that bedevil projects trying to get from a fundamental discovery to a discovery or to an intervention that improves human health. And by intervention, I should just mention, I don't mean just drugs here, I mean drug, device, behavioral intervention, any kind of intervention. For the most part, we're going to talk just today, just in the first few of these. We'll get into the fourth a little bit as well. But I mentioned some of these, the organizational problems, and the first one is something that is key to why you're here today. So those of you who don't know the rather remarkable story of this Assay Guidance Manual, it is one of the best examples I know of a forward-looking organization saying we need to do something different in order to change the way, change the outcome. That is, if we continue doing what we're doing, we'll get what we're getting. And so let's change that. And that organization was Eli Lilly. Back in the early 2000s, when a few of us at what is now NCATS had started the NIH Chemical Genomics Center. And one of the things that was evident to us was that the information that all of us had access to when we were at at each of the companies from whom each of us had come, and one of those people is Doug Auld, who's sitting in the back, and he'll be talking to you later. It was clear that for investigators in the public sector, those data, the data of how to do what you're going to hear about today, just was not available. It was available to us in the private sector, but when we left, it wasn't available anymore. And so through the work of Bill Chin and Siddha Satampalam and others, and Doug and Jim and Glace on our side, among others, there was a decision to take the internal Lilly drug development manual, or what was then called the quantitative biology manual, take out all the Lilly references and put it on our website. Now you can imagine when I heard this, I was more than a little astounded. This was not what you read about in the press about how pharma is supposed to behave. But that's exactly the point, that there were people who said, well, gosh, that's not what you hear isn't representative in the first place, but what we're going to do here is to say that good science should not be competitive. Good science benefits everybody. So let's release all these data. And from that, over the last now 12, 13, 14 years, has come the Assay Guidance Manual. And it all fits into this idea of data transparency as well as education and training down there and collaborative structures. So this is the part of the organization that this particular activity comes from. NCATS, just to finish up on NCATS just for a second, NCATS, like a lot of translation organizations, has a lot of activity in the clinical space. Probably 3/4 of what we do is in the clinical space, budgetarily. About 15% is in the preclinical space, and the part of the organization that Nathan works in is in that part, the NCATS Chemical Genomics Center, and the other parts are in re-engineering efforts, things like the tissue chip program that you may have heard of. So these assay guidance workshops for high-throughput screening and lead discovery interestingly started because of public demand. This is a good reason to start something like this. It's not often the case, right? Very often these kinds of seminars happen because a particular organization that runs seminars needs to make some money out of running a seminar, or a professional organization does. But this came from the fact that so many people, and I hope Nathan will show you the statistics, it's just extraordinary usage statistics on this manual, showing us just how great the need is, far greater than I anticipated, frankly. I think most of us anticipated. That there was a desire to have a voiceover. You know, if you're new to this area, just reading a textbook can be really hard. It doesn't matter whether you're like me, trying to learn how to, believe it or not, cook at a ripe old age that I am. I can read a cookbook, but I don't understand anything about how it says. It's only when watching old Julia Child videos that I can actually figure out how to do this and to talk to experts. And so the local expert equivalent to Julia Child is Nathan Cousins and others. Doesn't quite have the Julia Child voice, but other characteristics are similar, I'm sure you'll find. But the point is that you have to be able to talk to somebody who's an expert in the field to tell you, to explain to you in English how to do this. And so we started this a number of years ago now, and it's been increasing in time over the last few years. And this is just a picture of one of them. A number of the folks are in this picture. You can always pick out Tom Chung in these things because of that mustache. So there he is in the back row. So there's TC's in the back row. You'll hear from him later. But we really have had an enormously positive response to this. And so, as Nathan said, what we're trying to figure out how to do is to promulgate this as broadly as possible. So our videotaping this today is part of that mission. So here are the current statistics. I'm sure you've read each one of these 1,230 pages. If you haven't, that's part of the task for the next 10 hours. You will be reading each one of those. Just kidding. So there are now 43 chapters, and this shows you the retrieval statistics, which are just extraordinary. When we started this, we were surprised to see even thousands, even tens of thousands of unique users and retrievals per quarter of individual chapters. And you'll notice this is, yes, you're reading those numbers right. This is 120,000 retrievals in a single quarter. And from hundreds of different countries, all over the world. And one of the things we did with this is we decided that this was going to be freely available content. It's one of the things that does not characterize this world so much. So to have freely available content and actively updated content, much like those of you who are old enough to remember this, the original Maniatis molecular biology manual, or the current protocols in molecular biology, what have you. This is really thought of in the same way. There's growing from its original source, founding members, founding editors, and contributors at Lilly and at NIH. There's now an international editorial board of people who both edit these and solicit chapters. And Siddha Satampalam has been the editor for most of this time, and Nathan is taking over that responsibility now. So this is one of my favorite sayings, which really characterizes a lot of translation, and it really characterizes what you're going to experience today. That is, in the translational world, there is actually a lot known. Unfortunately, it just doesn't happen to be resident in any single person's brain. It's one of the reasons why translation is a team sport, but it also puts makes even more important than in other areas of science this concept of dissemination, of sharing. So much is known, but unfortunately in different heads. One of my favorite sayings from a bacteriologist of the last century. And this is what I imagine all of you undergoing today. So this here is you. This is Nathan and the other instructors here with his teacher's hat here on the top. I imagine this is Siddha here in the priest's robes, now making sure that the true light of the Assay Guidance Manual is carried on. And some of the other instructors over here, this woman has a book on her head, and that is meant to connote that we hope each of you will go out of here, print the entire Assay Guidance Manual, and walk around with it on your head. That has been shown to increase diffusion into the cortex. So it's kind of like the old Southern blots, you know, you have to put it on the top of your head. And this operation, believe it or not, is called the stone operation. There was a belief that insanity actually came from a stone in the brain. So what this individual is doing is drilling a hole in this person's head in order to remove that stone. So we look forward to having many stones released from you over the course of the day, but we hope this will not happen to you. Believe it or not, this is my new puppy who we just got last week. And he came up to visit the lab last week, and he was attracted immediately, as everyone is, to the Assay Guidance Manual. And then on a page, I understand on page 714, he fell asleep. He just couldn't stand it any longer. So one last thing while I show you the how to connect with us, although you'll be hearing about this later. You know, we think a lot around here about how to develop new ways to do the translational process better, new kinds of tools and technologies, some of them quite fancy, like the kinds of robotics that we and others work with, or the tissue chip platform, and some not so much, not so fancy, and then to disseminate their demonstrate their utility in individual use cases. So we do a lot of projects using those new technologies, and you'll hear about some of those examples, I think, today. But then we think an awful lot about dissemination. If we develop new ways of doing things and demonstrate that they're useful in individual use cases, but nobody knows about them, we haven't helped anybody, really. We haven't really changed the world the way all of us want to do. And so we spend a lot of time thinking about dissemination. And when you ask yourself, what is the most easily disseminated tool or technology? That has to be knowledge. Has to be knowledge. And that's one of the reasons that we put so much effort on what you're here for today. That knowledge is now transportable electronically and instantaneously. If we can increase the level of knowledge and practice in this field, we will have really revolutionized the success rate, we think, of this process. But it's also evident that this process of dissemination requires, like everything else NCATS does, innovation. It's not clear how to get this kind of information out to all the people who need it. And we've certainly managed to succeed in getting the paper version of this to hundreds of thousands of people. But one of the things that we continue to work on is how, through the most modern efforts of communication, how can we make this series available to as many people, accessible to as many people as possible. And you'll be seeing some of that today. I must say, I did ask Nathan and his colleagues whether it was possible to condense the 1,200 pages of the Assay Guidance Manual to a 140-character tweet. They're still working on that, as I understand. But he may announce that at the end. So I just want to welcome you, give you context of where you are and why what you're doing is so important to our mission, and that we hope you will go out and spread the word, not only about as we like to say, smarter science, but also encourage your colleagues to take advantage of this content, which we're eager to have reach as many people as we possibly can. So thank you and welcome.